Okay, good morning everyone. So today I will discuss about the process of glycolysis. We will talk about the process of glycolysis, energy investment phase first. So first of all, we need to memorize the glycolysis by using the tips, which is PIPLI. P-I-P-L-I. So how we use the tips, I will explain to you later. Okay. First, we will take the glucose, and the glucose will take the inorganic phosphate from the ATP to form the glucose phosphate. This process we need to use the enzyme, which is hexokinase. And the process for this we name it as phosphorylation. Phosphorylation P. So we use the word P here. Then to form the glucose 6-phosphate. Now we move on to the second step, which is isomerization. Glucose 6-phosphate will undergo isomerization to form fructose 6-phosphate. And then fructose 6-phosphate will undergo phosphorylation again to form the fructose 1,6-bisphosphate. We move on to the fourth step, which is lysis. Fructose 1,6-bisphosphate will undergo lysis process to form G3P and DHAP. Then the DHAP will undergo isomerization process to form G3P. So now you will obtain 2 G3P here. Now we move on to the energy payoff phase. We enter the energy payoff phase by memorize the tips using this. OSIRS. OSIRS. So how we use the tips, I will explain to you. Okay, now the G3P molecule will undergo oxidation to donate 2 H atom and undergo oxidation. Then the G3P molecule will combine with the inorganic phosphate to form 1,3-bisphosphate glycerate. Then the 1,3-bisphosphate glycerate will undergo substrate level phosphorylation to form 3-phosphoglycerate. The 3-phosphoglycerate will then undergo isomerization to form 2-phosphoglycerate. The 2-phosphoglycerate will remove the H2O molecule from itself to form the phosphoenolpyruvate. We can also call it as PEP. Okay, PEP. Then we move on to the last step, the phosphoenolpyruvate, which is also called it as PEP, will undergo substrate level phosphorylation to form the pyruvate. Okay, after the glycolysis process, so now we will move on to the link reaction. For the link reaction will occur oxidative decarboxylation. Oxidative means oxidation. Decarboxylation means removal of 1 carbon dioxide molecule. Okay, so now we will take the product from the glycolysis, which is pyruvate. The pyruvate will release 2 hydrogen atoms and undergo oxidation. The 2 hydrogen atoms will combine with NAD+ to form NADH plus H+. Then the pyruvate will undergo decarboxylation, removal of 1 carbon dioxide molecule. So the pyruvate is the 3 carbon atom molecule. It will change to the acetyl group, which is 2 carbon atom molecule. The acetyl group will combine with coenzyme A to form acetyl coenzyme A.
everyone. Today I'm going to talk about oxidative phosphorylation. It is a final stage in aerobic respiration, which consists of two parts, which is ETC, the electron transport chain, and chemiosmosis. And oxidative phosphorylation occurs at the internal membrane of mitochondria, which is the cristae. It needs two electron carrier, which is NADPH and FADPH2. And these two molecules will be oxidized and transfer electron throughout the membrane, and eventually will produce ATP. So the first process starts with NADPH2, will transfer electron from NADPH to NADPH dehydrogenase. And we can know, and as for your information, the transfer of electron will produce energy. So the transfer of NADPH to NADPH dehydrogenase will produce energy, and this energy will pump H+ ion throughout the internal membrane, from the matrix to the internal membrane space. And the electron will continue pass from NADPH dehydrogenase to succinate dehydrogenase to ubiquinone to cytochrome C reductase to cytochrome C, and to lastly cytochrome C oxidase. However, this is not the final electron acceptor. The final electron acceptor is oxygen. So oxygen will get the electron from NADPH and plus with H+ ion, and eventually will produce water. So that's the ETC part. The function of ETC is to pump hydrogen ion from the matrix to the intermembrane space. So let's pass to the next stage, which is chemiosmosis. Chemiosmosis is the final producing the ATP, the process that produces ATP. So as you know that H+ ion is transferred from matrix to intermembrane space. It will let the intermembrane space, the concentration of H+ ion to increase. And if the H+ ion is increased, it will try to move from high concentration to a low concentration. However, throughout the intermembrane, throughout the cristae, only as ATP synthase will able to let H+ ion to move across. So the moving across from the intermembrane space of H+ ion through the membrane and eventually go back to matrix will produce energy. And this energy will use to phosphorylate ADP plus P inorganic to become ATP, which is the final product that we want. And for your information, the pumping of H+ ion only occurs at three places, which is NADPH dehydrogenase, cytochrome C reductase, and cytochrome C oxidase. And we all know that the electron will transfer from less electronegative atom to a more electronegative atom. And FADPH2 is more electronegative than NADPH dehydrogenase. Therefore, in the first step, it will not transfer electron from FADPH to NADPH dehydrogenase. Therefore, the pathway will be shortened. It will only transfer electron from FADPH2 to succinate dehydrogenase, and it will only pass through cytochrome C reductase and cytochrome C oxidase. Therefore, the hydrogen ion will be pumped twice only. So if one hydrogen ion will produce one ATP, then we can conclude that for every one molecule of NADPH will produce 3 ATP, while FADPH2 will produce only 2 ATP. So, and that's the whole process of oxidative phosphorylation, which includes the transfer electron, pumping of hydrogen ions, and pumping back, and the movement back to for the hydrogen ion to the matrix, and eventually produce ATP. And that's all. Thank you very much.
after the process that has been mentioned before, which is link reactions, link reaction, glycolysis, and also the Krebs cycles, we're going to talk about the ATP production in complete oxidations if 1 mole of glucose has been oxidized completely. So now we can say that the ATP can be produced through the oxidative phosphorylation or also the substrate level phosphorylation. Now we're going to look into the oxidative phosphorylation first. So firstly, we know that 2-NADH will be produced from the glycolysis because of the process that 2G3P will be converted into 1,3-BPG. In this process, we will release 2-NADH. Thus, as we know, for the context, each NADH will release 3 ATP. So now we can say that 2-NADH will produce 6 ATP in the process. And after this, we're going to move into the link reactions. Link reaction will also produce 2-NADH by the process of pyruvate has been converted into the acetyl-CoA. So we know this will also produce 6 ATP. And then we're going to move into the Krebs cycles. Krebs cycle is a long reactions, and thus there got a lot of process that will produce NADH. So firstly, it is isocitrate to the alpha ketoglutarate. And each alpha isocitrate and to the alpha ketoglutarate, it will produce 1 NADH, and thus 2 will produce 2 NADH. And after this, we will say that the alpha ketoglutarate will be converted into succinyl-CoA. And this process will also produce 6 ATP, same as the process above, which is 6 ATP also. And eventually, we got the 2 malase be converted into the oxaloacetate and will produce 6 ATP also. And lastly, we are going to move into the Krebs cycles of they will produce FADH2, which is the process that 2 succinates will be converted into 2 fumarates. And for context, each FADH2 will be released to ATP. So we can know that all of the oxidative phosphorylation has released 6 times 1, 2, 3, 4, 5, 5 plus 2 times 2 ATP, and then it will have 34 ATP. And after this, we are going to move into the substrate level phosphorylation. So firstly, we can look into this, which is 2 G3Ps will be produced by the Krebs cycle, which is the process of 2 succinyl-CoA be converted into 2 succinate. And then we can say that 2 GTPs will release 2 ATP also. Lastly, we are moving to the glycolysis that will produce 2 ATP. Actually, it will produce 4 ATP, but it will also use up 2 ATPs, and thus we will left 2 ATP only after the process of glycolysis. So now we can say that in the oxidative phosphorylation, we got 34 ATP. While in the substrate level phosphorylation, we got 4 ATP. So we can add up it, and we say that the complete oxidations of ATP productions in a complete oxidations will release 38 ATP. After listening to the other polymer's explanation, let us move to another structural question. Figure shows one type of cellular respiration process. This is the diagram that shown by the question. The first question is what is the process A, R, and S? First, process R is the process that glucose convert to the 2-pyruvate acid. They produce the 2NAD plus convert to the 2NADH, 2ADP plus 2P convert to the 2ATP plus water. This process A is the glycolysis. The process R is the 2-pyruvate acid is converted to process R and process S. First, process R has some bacteria and mammal muscle. Their process R is occur in some bacteria and mammal muscle. That's the process R is the lactic acid fermentation. This is because the lactic acid only can be found in the bacteria of the mama muscle. Next, the process S is occur in the yeast. As we know that the yeast will produce the ethanol when he's carry out the anaerobic respiration. So the process S is the ethanol fermentation. B question is what is the type of cellular respiration as above? The type of cellular respiration above is the anaerobic respiration. This is because this process is produced that occur in the yeast and the mama muscle without the oxygen. So this type of cellular respiration is the anaerobic respiration. The question C is what happened to the primary acid in available oxygen? The primary acid will enter the mitochondrion and convert to acetate CoA. Let's move to the question D. State the difference between process R and S. First, process R is the lactic acid fermentation and the process S is the ethanol fermentation. First, process R produces the lactic acid while the process S produces the ethanol. Second, process R also not release the carbon dioxide while the process S release the carbon dioxide. Question E. What is the importance of cellular respiration in our daily life? First, the cellular respiration can produce cheese and yogurt in the dairy industry, which use the lactic acid produced by the cellular respiration. Second, the wild beverage and alcohol production also can produce by the cellular respiration by using the ethanol. Thank you.